Tell your parents to pay the bill. All right. So what we got is Sodaria Fimicola we're dealing with today. I have pre-written some notes for you. All right. So what we have is, we have brown and mutant, okay, fungi. These guys, of course, are decomposers. They recycle nutrients for ecosystems. And basically, they grow from an ascospore. Just like you think about a spore, okay, from mushroom. They're in the fungi family, all right, fungi. These individual ascospores, and the reason why we call them that, I'll talk about in a second, produce adult fungi that grow in their medium. And interesting about them, they call them mycelium, even though I probably didn't mycelium, okay? Any case, they're adult, so the tan and brown Sodaria Fimicola grow from a single spore, which happens to be an N number, meaning it was a haploid. And they grow into adults as haploids. That is really odd, okay, in the biological world. Most things have to become a zygote through sex reproduction because why? Sex reproduction gives you diversity, okay? This doesn't give you the same diversity, but fungi have been amazingly what? Good at surviving in its medium, so they've lived the test of time this way. Now, interesting enough, what we're going to take is the tan, the mutant-colored Sodaria Fimicola, and we're going to mate it with the brown. Okay? How are we going to do that? Well, I'm going to give you a culture, okay, made up of agar. Okay, it's a sugary-based food that's very, very a gel-like substance, kind of like Jell-O. Okay, so it's going to sit very firmly into the petri dish, and we're going to take our petri dish, looking down at it, and cut it into quadrants. And essentially, I'm going to give you a little piece of the tan and brown Sodaria Fimicola that I've been waiting for to come in, okay? And you're going to take a petri dish, okay? I'm going to give you a little slice of it. So here's the petri dish looking from the side. Here is the agar, okay? And then, of course, we're going to have the fungi on it. Well, I'm going to cut that into a little piece, and you're going to take the mycelium side up and put it right into that quadrant. This could be the tan, and we'll put the tan opposite of each other. Okay? Then I'll get you, there's another, okay, agar plate, okay, that has the tan, has the brown. I'll draw the brown. I know it's the wrong color, okay, but cut a little piece, and then you're going to stick that into the mycelium side into. And so notice what I'm doing. We're going to put a little piece here. And so what I'm doing, or what you'll be doing when you culture these plates, is you'll be putting the two tan on these opposite quadrants and the brown on the other. Okay? The mutant here and the wild type there. And what's going to happen, obviously, is the fungus is going to grow outward in all directions. And interesting enough, a beautiful picture of that. Cade's, this is an example of what it looked like after, I forgot how long it was. It's a beautiful picture here of the tan and the brown. Okay, so here's the, I think the brown and the tan, and you can see some lines. Okay, that's not actually the lines that you drew, although they're there. Okay, so some of that's there, but this is where you're going to have those dihybrids. Okay, and these dark areas are where the mature zygote that results from two myceliums growing into each other for the first time creates a diploid organism. Okay, and I drew the chromosomes here. So they have single, well, the chromosome that we're studying, or that chromosome that has that allele, their N number, and then here they become, they eventually have homologous chromosomes here. We call that a hybrid. And it's these hybrids at this line, or this junction here, that we're after. And I'll tell you why. Because the color, the tan and the brown Sodaria Fimicolas, they're colored because of the color of their spores. Remember, we call these an ascospore. They're ascospores because these spores result, okay, or the color of the fungi is due to the color of the spores that exist in what we call an ascus. I can't spell, I guess. Ascus. So this is called an ascus, okay? And that's why they call them ascopores. All right? Because they are, and I think I may have killed that as well in terms of spelling, but the bottom line is you have 8 of these spores. Now, when do they result? Well, once a zygote is made, okay, and of course, that's going to happen where they grow and meet each other, correct? So that's why that divider line is drawn. We're going to take samples from this area here, from these quadrants, the lining the quadrants, because that's where the tan and the brown meet, and that's where the micellums will hit to create the zygote or the dihybrid right here. So we're going to take those samples, okay? And we're going to look at these spores. Remember, the ascus that holds the ascopores are only made when there is a zygote. So when these guys do undergo sexual reproduction, okay, which occurs right here, okay, remember, they can live their entire life until they reproduce to make a zygote. So once you are a zygote, they can make something called a perithesium. A perithesium is a container, like a fruiting body, that holds these ascus, or asci. I guess that's an asci, and ascus is many. Asci is many, whatever. The point is, we can look at the color of the spores to determine what kind of crossover is occurring. Obviously, when you breed these, you're going to have a color that holds both. You're going to have some tan, and you're going to have some brown ascopores in an ascus. What I'm telling you is that when we take samples along these lines that have the dihybrids, that have a mixture of the color for the what? Tan and the brown color of the ascopores that are in these sacs called ascus, we can actually look and see for whether or not there was reproduction that made hybrids, but more importantly, based on the color of the spores, we can determine what kind of crossover has occurred. That's the cool part. That's what this is all about. Recombinance is all about mapping how close these guys might be, or how close they are to the centromere anyway. So let me talk about that. So it seems a little bit confusing, but essentially, single chromosome, or in this case, haploid individuals make a zygote, okay? Brown and tan. The color is due to this, basically the color of their ascopores that are in their little sacs called ascus. When they come together, there's going to be parts of these ascus that have both brown and black. That's how we know we have hybrids. But the color combinations in these ascus will tell us something about the crossover. Let me explain that. That's the part that's the most complicated, because what we're after in this lab is, of course, percent recombination. And then we kind of want to sort of map this chromosome to what we call the centromere, the middle part, okay? So let me try to make some sense to this, all right? All right, so in any case, let me show you, because what's cool is we're going to put these under a microscope to look at these. So in any case, let's take a look at what we have. 2 ends come together. Well, that's happening here. Hey, the what? The tan is going to grow out in this food. The brown is going to grow out. And eventually, as they expand, they're eventually going to touch each other. That's where the sexual reproduction occurs. Okay, so that's what this means. N plus N, all of a sudden you make a zygote. Okay. Now, what do you think this means with the 2N duplicating here?
Meiosis.
Okay, and what's happening? We're getting ready for mitosis. What is this talking about?
Interphase.
Yeah, and what are we talking about in the growth cycle?
The S phase.
Yes, we got to duplicate our chromosomes. That's what they're showing us in this picture. And then we are in prophase. And then we know in meiosis there's 2 halvings. Because think about it. If you've got a 2N number right here, let's pretend, I know it's not right, but let's pretend that this fungus has a 2N number equal to. What does that mean? Now we're 46, so we have actually how many chromosomes? Our 2N number is
23.
Okay, no. Our 2n number is 46. Our n number is 23. Our sperm has 23 chromosomes. Our eggs for females have 23. Those are your haploids. So let's pretend that the Sodaria Fimicola, let's pretend it's not true, is a 2n number. And 2n number equals 2, just to make this simplify. Okay? That means each n number is 1. Okay? Now, so in any case, you're going to duplicate your chromosomes. So if a 2 number equals 2, if I said draw the prophase of a nucleus, which means they start to be coiled up, of a 2n number, a 2n equals 2 number, how many chromosomes do you draw? 4, correct. And what do they look like when they're in meiosis? So word that starts with a T. What do they look like? Well, if it's in meiosis, I'm going to have 2 of these. And I'm going to have, Christmas, 2 of the what? But it won't look like that, will it? What will that look like? It's going to be in a tetrad. Good. So let me show it the way I think it looks better. I have one connected to the centromere. I have another connected to the centromere. And then I'm going to have what? Whoop. What do we got going on here? Crossover. That's a tetrad. Okay? All right, now I'm showing crossover. Now, so if you notice, getting back to this, we duplicate our DNA. That's where helicase comes in, unzips, topoisomerase cuts. All right? Then we got what? We got single-stranded binding proteins, right? Prevent the unzipping. We have the DNA, we have primase put in RNA. We build from the 3 DNA polymerase. We have a lagging leading strand, all the stuff you've done with the voiceovers, which I'll collect today. Okay? And then, of course, all right, now we have it. Because remember, if I'm trying to make an ascopore, think about it for a second. If I'm trying to make an ascopore, and it has, what does an ascopore have? How many chromosomes? 1. We're starting with 4. Now, whether it's Sodaria Fimicola or human beings or chimpanzees or protists or bacteria, okay? Although bacteria doesn't do meiosis, but that's a bad example. But the point I'm trying to make is we duplicate, we have double the number of chromosomes. To get down to the haploid number, doesn't make sense we have to have twice. And you guys started that this summer. You have to have twice. We're starting with a 2n number. If this is mitosis, we do what? Have once. Because we duplicate this with 4, and then we'd have once to be back to 2. But because it's meiosis, we're starting with a 4n number, really, after this, and then we have, I would put 4n right here. Okay? This is someone else's drawing. But I still like it. And then here I'd have, and have again. And notice, at the end of meiosis, I have 4 daughter cells. Each with what?
2.
Each with 1 chromosome if I'm using the model that 2n equals 2. I know Sodaria Fimicola does not have 2, but I'm just keeping it simple. You with me? Good. And then 1 extra step. This is different. They undergo mitosis to duplicate that one, so that's where you get your 8. So party people, where does the color come from? The color comes from the individual spores. Okay? Individual spores. All right. So let me show you what you guys are going to be looking at after we culture these guys, if they ever come in. But it's such a great lab, I'm going to keep fighting for it. Okay? Because I watch it. It's really cool. All right? So, by the way, here's the life cycle. Here's an ascus. They discharge their spores. Hey, they grow into a filament or a mycelium, which is basically an adult. Okay? Here's my mycelium, grows with its food. All of a sudden, 2 adults touch, which are now going to make a diploid, which is the zygote. It's the zygote after fertilization that starts making these what? These ascus, or I should say, these packets of 8. They go into this mature perithesium, which basically will shoot these things out at the end. We're going to start looking at these to see what kind of mating has occurred, what kind of crossover could have occurred. Let me explain. Okay? Now this looks very confusing. Okay? Let's talk about this for a second. All right? So, where are we starting? Let's talk about no crossover. Notice, I've got a tan, I got a brown. Notice I'm starting with 4 chromosomes. Okay, and in that tetrad. How do I have, if 2n equals 2, how do I have 4? I had the duplication in the S phase. Right? That's what this is. They duplicated each other. We were here, and all of a sudden there's duplication. I'm not showing crossover here, so in this drawing, we have this. And you should know that the what? The black and the blue. If it's blue, if it's purple, I have no idea what color. Okay? But those guys are sister chromatins. Duplicates of each other. Homologous to each other, duplicates of each other. Okay. Now, so we have 4. All right. Meiosis One. Okay? Now, what we have here, and it's not showing it very well, but so this is preparation of Meiosis One, and by the end of Meiosis Two, this part kind of is not very clear, but at the end of meiosis, we have 4 daughter cells. Each one of these guys is produce what? Well, this holds the tan what? Color in its chromosome, so it's going to make 2 tan ascopores. And because there's an extra step where they duplicate it by mitosis, you make what? 4 tan, and for the opposite reason, during this becomes a single one, okay, at the end of meiosis 2, they duplicate that. And so if you have no crossover, we will see in our microscopes ascopores with 4 stacked up that are tan, and then 4 black, or vice versa. So if we go in our microscopes and pick samples right at the edge, we know we're in the right area if we get ascopores that have 4 and 4. And it can be top or bottom based upon, hey, where this is in the organism. And that we would count that as a hybrid. And we would count this as an individual that mated, but there's no crossover here. Okay? All right. So we'll actually see that, and I'll show you pictures and example. Okay, so what about crossover? Because this is all about how much crossover occurs to get frequency of recombinance. Now, this is different because we're only looking at one allele here, but we can still do this. Okay, so here comes that tetrad. Okay? We're right here, and then it was duplication. We're in the tetrad. Now there's crossover. So what happened was, only ones that cross over are the what? The ones in the what? The ones on the inside, correct? We talked about 50% recombinance. That's the highest you can get. That means that these guys cross over all the time. Okay, now, so here's our tetrad, and there's some crossover, which means due to a crossover event, okay, we broke the DNA at the phosphodiester linkage, and new DNA was transferred. And so now you brought the brown allele, positive wild type, over here, and the tan over here. Okay? Assuming that that gene is farther enough away from what we call the centromere. Look, this little part right here is called the centromere, if we forget about it. And the crossover event occurs from that part, from that centromere. Okay, so what happens here? Okay, well, you're going to take this and eventually make 1 chromosome in 1 ascopore, or at least almost. This becomes 1 here. Remember, we're basically going to have twice. Have twice, you get the individual 4 daughter cells that have the individual haploid. 2 is D. 2 is diploid. There's 4 here because of the duplication in the S phase. Now, this of course is going to make what? In mitosis, 2 tan. Oh, this has the what? Gene for brown. You make 2 brown. This has the gene for what? to tan, and then to black. And depending upon, you know, how this lines up, you can get all of these different combinations. And so when you look in your microscope and you pick from those areas, you'll count recombinant, 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 parental. And you'll count total amount of hybrids, ascus. Every ascus is a hybrid, okay? And you count that. So you'll be able to see the group of spores, and we'll be able to really physically see the inside of crossover right there, okay? Really cool lab. It's a little touchy to find, and people get frustrated, but I help you with it. And when you get it, it's really cool. And to show you the actual pictures, okay? This one I get off the net, okay? But just so you know, there's an example of a what? Recombinant. Here's a picture of parental. No crossover. Okay? So, recombinant. Recombinant. And you'll count all the ones that you can fully see. Now, these are actually pictures I've taken from previous years. Okay? And I blow that up, and you can see. All right? Hey, there's 4 in a row. I know that's a parental. Okay? Hey, too dark, too light, too dark, too light, recombinant. Okay, so really cool. Too light, too dark, recombinant. And then as you focus, 4 in a row, you definitely know. Okay, and how do you know that you have hybrids? You'll know because if you have a hybrid, if you don't have a hybrid, they'll be either all black or all tan, and you're in the wrong area. Okay, and I might have some more pictures from previous years. Nope. Any case, that's the lab. And then, of course, the last thing that we're going to do is we're going to take the crossover of the offspring and we'll divide by 2, okay, because only half of them can cross over correctly. All right, we'll talk about that. All right, and then it will be an activity. It won't be a full write-up, but a really interesting one in terms of getting some insight to recombinants. Hard to do recombination labs. Okay. All right, so that is the lab. I'm going to give you guys a 10-minute break while I am going to email everyone at home. I'm going to email your graded DNA experiment form that you had a couple of weeks ago. Sorry for the delay in that. I'm going to give that back, a form. It was a handwritten form. Okay, so take your 10-minute break. I'll let you know when I'm ready to join back up and reviewing that form.